Hello everyone, I am Shriyansh and today I will be presenting my work titled Drug Discovery in Time, Studying Binding Pocket Dynamics of REV-ERBβ for Rational Design of Small Molecule Modulators. Before going about explaining the scientific part of my talk, I would like to highlight that we all have clocks in our biology, and essentially, one of the most fundamental rhythms that are observed in physiology and behavior are circadian rhythms, which have a 24-hour cycle. These circadian rhythms in the physiology can be seen as the very commonly observed sleep-wake cycle wherein we are awake during the day and sleep during the night, and the associated intrinsic molecular adaptations and changes that regulate the metabolic cycles during the day and during the night. And this rhythmic pattern can be attributed to one of the most widely studied transcriptional translation feedback loop comprising some of the transcription factor known as lock and BMAL and other core clock proteins and closed clock genes known as REV-ERB, ROR, cry, and other clock control genes. Today, I'll be talking more about REV-ERB beta, which essentially works as a transcriptional repressor and represses the clock gene BMAL1 and other target genes by recruitment of NCOR, nuclear co-repressor peptide, and HTAG3. This regulation of REV-ERB has been implicated in several of the disorders wherein it is upregulated in some while downregulated in the others. Now, REV-ERB has been studied as a drug target, and efforts have been made to synthesize small molecules that can be given as an agonist or an antagonist, which in turn would regulate binding of NCOR with the ligand binding domain and regulating the transcriptional repression. Now, these agonists and antagonists had been developed using thread-based screens and gene reporter assays, and the structural mechanisms of how a small molecule behaves as an agonist or a molecule behaves as an antagonist remained unknown. Based on this lacunae, we asked two questions wherein we asked what is the conformational landscape of the ligand binding pocket of REV beta in different states and what actually constitutes as a structural basis of a small molecule acting as an agonist or an antagonist. To answer the questions, we first looked at the crystal structures for AVRB beta ligand binding domain, performed the simulations in EPO and heme bound form, then docked the small molecules that have been experimentally discovered, performed the simulations in presence of agonist and antagonist, and look at the NCR peptide binding dynamics in presence of agonist and an antagonist. To very briefly explain the domain architecture of REV-ERB, so these REV-ERBs are act as constitutive transcriptional repressors because they lack the C-terminal AF12 domain, which is essential for recruitment of coactivator peptides, and because it is not there, these REV-ERB can only recruit NCOR and thus act as transcriptional repressors. Heme acts as endogenous ligand for REV-ERB beta and is critical for co-recruitment of NCOR peptide. The heme is coordinated through the iron center with cysteine on one side and histidine on the other. We took all the structures available in the PDB for REV-ERB beta ligand binding domain and clustered them, and what we could see that the ligand bound or the heme bound structures formed a cluster which was different from the EPO form of the structure, and we superimposed them, and then we could visually see that the helix 11 had a different conformation in EPO state as compared to the bound state. Next, we performed molecular dynamic simulations for AVRV beta ligand binding domain in EPO and heme bound form. Upon running the simulations, we could see that heme could stabilize binding pocket dynamics at helix 3 and helix 11, and in absence of heme, the pocket closed and it took conformation similar to the EPO form. Next, we performed molecular docking studies of experimentally discovered agonists and antagonists to REV-ERBβ ligand binding domain. We performed molecular docking studies for agonists and antagonists in NCORID1 and NCORID2 peptide bound states to REV-ERBβ ligand binding domain. We took these docked complexes of agonist and antagonist and ran simulations to understand the dynamics of NC or ID1 peptide and ID2 peptide. In ID1 peptide, we could see a higher fluctuation at the N terminal of ID1 peptide in presence of antagonist but not an agonist, and an overall higher RMSF for protein in presence of an antagonist. For NC or ID2 peptide, we could see that the overall RMSF for the NCR peptide in presence of an antagonist was higher as compared to the agonist. Tried to characterize the interactions of ID1 peptide in agonist and antagonist bound state and could see differences at the helix 11. Next, we also tried to characterize the distance between the helix 3 and helix 11 and saw that in presence of an antagonist, the distance increased after approximately 50 nanoseconds. Overall, we could see a differential binding of antagonist and agonist and differential dynamics of ID1 peptide in presence of an antagonist and agonist. The future work of this study is to perform comparative study on alpha isoform, perform structural based virtual screening, and validate these experiments in the lab. I would like to thank all the lab members and all the funding bodies for this work. Please feel free to check out our lab website and read more about this work, which is now available as a pre-printed bioarchive. Thank you.